değerli hocalarım, değerli meslektaşlarım, sevgili arkadaşlar. Türkiye Üreme Sağlığı ve İnfertilite Derneği ile Klinik Embriyoloji Derneği'nin ortaklaşa düzenlediği bir webinarla tekrar karşınızdayız. Bizlere bu imkanı, Klinik Embriyoloji Derneği olarak bu imkanı sunan başta saygıdeğer dernek başkanımız Ahmet Zeki Işık hocama ve nezdinde bütün YK üyelerine tekrar teşekkürlerimizi sunuyorum dernek yönetim kurulumuz olarak. Bu akşamki konuğumuz Doktor Denny SAKKAS, konumuz Non-invasive embriyo seçimi. Acaba biyopsinin yerini alacak mı? Çok ilginç bir konu. Günümüzün en ilginç, en önde gelen araştırmacıların da odaklandığı konulardan bir tanesi. Kendisi bizlere hem bu alandaki çok tecrübeli çalışmaları, yaygın çalışmaları üzerinde bir derleme yapacak, sunacak. Ben kendisine mikrofonu vermeden önce kısa bir tanıtım yapmak istiyorum bu konuşmacılarında. Her ne kadar birçoğumuz derneğimiz vasıtasıyla da tanıyor olsak da Doktor Denny SAKKAS lisans eğitimini Avustralya'da Melbourne Üniversitesi'nde aldı. Doktorasını yine Avustralya'da Monash Üniversitesi'nde tamamladıktan sonra başta Amerika Birleşik Devletleri, İsviçre ve Avrupa'da pek çok tüp bebek ünitelerinin, laboratuvarlarının hem bilimsel direktörlüğünü hem de laboratuvar direktörlüğünü yaptı. Yani kendisi klinik embriyoloji alanında, üreme tıbbı alanında, sağlığı alanında çok detaylı, ciddi, uzun süreli tecrübelere sahip. Uluslararası camiada kendisini son derece yakından tanıyoruz. Başarılı çalışmalarını tanıyoruz. Döllenme, erken embriyo gelişimi ve erkek kısırlığı alanında 200'ün üzerinde hakemli dergilerde yayınlanmış makaleleri, kitap bölümü var kendisinin. Aynı zamanda bugün başta Human Reproduction Open olmak üzere alanımızda önde gelen 5 büyük derginin de yayın kurulunda görev alıyor. Şu anda kendisi Boston IVF'in bilimsel direktörü ve aynı zamanda Yale Üniversitesi'nin Yale Üniversite Fakültesi Kadın Hastalıkları Jinekoloji bölümünde doçent olarak ders veriyor, görev yapıyor. Çok da teşekkür ediyoruz kendisine gerçekten bizleri kırmadığı için, bu akşam birlikte olduğumuz için. Doktor SAKKAS aynı zamanda Klinik Embriyoloji Derneği'nin de onur üyesidir. Bundan dolayı da çok mutluyuz. Kendisi Türkiye'ye çok sık geldi gitti son dönemlerde. Hani yarı Türk sayılı diyebiliriz. Umarım bundan sonra kendisine bunları da tabii tercüme ederek anlatacağım. Denny, can you hear me?
Yes, okay. I can just hear you, not very clearly.
Excellent. So I'll just try to explain your previous research about the area and your short biography. And I'd like to leave the stage to you. And again, thanks to you for accepting the invitation and to be with us tonight.
Thank you everybody for joining. I know it's 9 o'clock in the evening in Istanbul or Turkey, so I hope you're all staying safe and taking care. It's a very strange situation for everybody, so it's the same for everybody. So today I'd like to give a talk about some work that I and many others in the area have been working on for many, many years. And it's about the non-invasive selection of embryos. And the main question I'd like to pose is that, will it replace biopsy or PGT analysis?
Excellent. Yes, you may go on.
Okay. So the biggest problem we have in actually selecting which embryo is the best is actually this gap here, that we can pick a good embryo, we can transfer those good embryos, but our problem is actually selecting the best embryo and knowing which embryo to transfer. As I just mentioned, and I hope you saw this this before, but obviously not. This is what we're faced with. So this is the technology we now use to actually to pick the best embryo, where we biopsy the embryo, we remove 5 to 6 cells, and we're faced with a genetic analysis and a technique that is actually quite difficult. So what else can we do? We're lucky with embryology. We're lucky with embryology because we actually have access to a number of things with the embryo. We have access to the culture media that surrounds the embryo, and we also have access to actually looking at the embryo. So obviously we've been looking at embryos for many years, so we've become very good at that. So what I'm going to talk about is some of the technologies that allow us to actually access information of the embryo without touching it. And obviously one of the first ones is morphology and cleavage, or actually looking at time-lapse or morphokinetics. So the morphokinetics is in terms of time-lapse, and there are many different systems available, gave us the hope that with morphology and the timing of embryos, we will be able to do a lot of information. We'll be able to improve our pregnancy rates. Unfortunately, just that data in itself really didn't sort of change everything dramatically. And we're still, stuck with the idea whether the time-lapse systems really have helped us. But in the last year, we've had some key papers that have shown us that there is a future for time-lapse imaging assessment. And the reason this has changed this area of research is that we now are able to use artificial intelligence to look at the data that we get. We get a lot of data about embryo development, embryo cleavage, the types of embryos developing, and also we're able to use recognition software to look at these embryos. So what I'm going to show you are just some quick data about time-lapse, and this would be a whole lecture in itself, and I'm sure at some stage in the future you will have a lecture about this, but 3 key papers that have come out in the last year discussing time-lapse. So the first one was by Tran et al, and it's in collaboration with David Gardner's group in Australia, and they've shown us that deep learning could act as a predictive tool. And what they did is they took embryos, over 10,000 embryos, and they looked at their fate, so whether they were discarded, frozen, or whether they actually underwent fresh transfer. They used the information from the time-lapse system, and in this case, it was an embryoscope, and created very sophisticated mathematical models that allowed them to try to predict which embryos would actually give them a pregnancy. And what they did was create a curve that would actually give you a prediction of which embryos would actually, if in the algorithm, which embryos would be picked to actually go on and have a fetal heartbeat, or those that would not. The line in the center on the graph in the right-hand side gives you a 50-50 chance. And what is amazing is that this analysis allowed them to predict with about 93% accuracy which embryos would actually be the ones that would give you a fetal heartbeat. So this is actually amazing data, and we're obviously now looking forward to a clinical trial in this data. So this is data that was done on a known data set with some blind testing, but we think that this may actually be very productive and very efficient in terms of looking in the future. What about other systems? This is a paper that just came out recently, and it's a bit different from what we've seen before because it doesn't look at actually time-lapse imaging. It actually is looking at a single shot of an embryo, a single static image. And it's proved very interesting because this is work done by Tex Vermilea and a company called Life Whisper, again from Australia. And what they did was that they looked at images of blastocysts and were able to predict to about 70% chance which embryo actually was going to be the viable or the non-viable. So I won't go into the whole paper, but this is a very interesting paper, and I think I would urge you to look at these types of papers. So it's very promising, the advent of artificial imaging, artificial intelligence, image recognition software, and allowing us to possibly predict embryo development and embryo viability. There was also another paper from a Brazilian group doing the artificial intelligence in collaboration with the Cornell group here in the US, looking at deep learning and mathematical modeling, but looking more at how we assess human blastocysts. So basically, they built a protocol where they could actually allow the microscope to actually predict which embryos would be the poor quality and the good quality blastocysts. And they compared this to actually a range of embryologists. So they developed this stalk system that they called, and this could actually predict with very good accuracy which embryos were poor blastocysts and which were good. And you could argue actually that it was better than actually the embryologist. So this is a prediction of against 5 or 6 embryologists, and actually predicting which were the good blastocysts, and that's the yellow highlighted area on the top. That's the embryos that were predicted as good by the software. And you can see that actually there was quite a range between the embryologists in actually being able to predict which were poor and fair embryos or blastocysts. So this data shows that actually you could use artificial intelligence to predict which is the good blastocyst or the poor blastocyst. When this was tested in relation to pregnancy, it wasn't as good at predicting successful pregnancy. So this still needed a bit of work, I think, in terms of predicting pregnancy also. But there are many groups, many companies now that are using this type of data and using time-lapse to actually create algorithms. And I think we will see much more in the future. There's one thing, though, that morphokinetics and morphology, as we know, does not tell us. So this is one of my favorite slides. So because what it tells you is that when you're looking at something, is you don't always get the full story. So here are three people, and some of them will be known to some of you. So Winston Churchill on the left, Roger Bannister in the second, and Jimmy Fix on the right-hand side. So the two on the right-hand side were obviously very fit people. They were athletes, very high-level athletes. Winston Churchill was the complete opposite. He liked to have a good drink and smoke cigars and wasn't your picture of an athlete. But you can guess if I tell you who lived the longest, because this is probably a loaded question. So you can see when I tell you this is how long people live, that just looking at people and getting some story about their lifestyle, let's say, will not tell you everything about the viability, let's say, here. So obviously, Winston Churchill here lived the longest, but you would not have guessed that if you knew all the information. So just by looking at embryos and maybe getting some morphokinetic history may not give us enough information to tell you this is the best embryo. And we know this already because our best PGT or genetic testing results from the best chromosomes, the best euploid embryos, only really give us about a 60–65% live birth rate. So we're still missing some data in actually assessing embryos. We've known for quite a while that if we understand the inside of the embryo, and in this case metabolism, we can actually look at the viability of an embryo. So I think in the future, what we may see is that understanding the metabolic imaging or metabolites that an embryo is using may give us actually more information. It may actually give us more information than we think it will. So we're looking at testing viability by looking at the metabolism of embryos. And many people in the past have done this, including people like Henry Lees, David Gardner, and even in 1980, Jean-Paul Renard and Yves Menizo used glucose uptake, for example, to measure embryo viability of bovine embryos. So what I'm going to talk about in the next few slides is a non-invasive system of testing the metabolism of embryos. And this system is a microscopy system, and it's called fluorescence lifetime imaging microscopy. And what FLIM does, fluorescence lifetime imaging microscopy, it measures the average time a molecule stays in an excited state before emitting a photon. And it's very good at measuring the metabolism of cellular function. And what it does, it allows us a novel non-invasive technology for embryo imaging. And interestingly, two of the metabolites or the molecules that it can measure are two key metabolites or factors that are involved in mitochondrial metabolism, NADH and FAD. So why would we use these? If many of you can remember your metabolism and your mitochondrial metabolism, you'll remember that both NADH and FAD are very important intermediates in the electron transport chain. NAD in particular acts as a shuttle for electrons during cellular respiration, while FAD is a coenzyme of oxidation reduction to replace NAD. And NAD and FAD are both key elements to cellular respiration and ATP production. So why am I telling you this? One, because NADH and FAD both can be measured by FLIM microscopy. And the most interesting thing is they do something that's called autofluoresce. So you don't actually have to add anything to a cell, or the embryo in our case, to be able to measure their fluorescence. So with using FLIM technology, or fluorescence lifetime imaging microscopy, we can actually measure the activities of these photons. And so how does this happen? So basically, under a very small laser source, the fluorescence lifetime imaging microscope will excite the fluorophores, and these take time to de-excite. So they take time to de-excite, and this time can actually be measured and actually plotted in a histogram. So what we can do is actually excite both NAD and FAD, and we can actually get the time of arrival. We can get a lot of information about how they're functioning on the mitochondria. So this is an example of how we get metabolic parameters for NADH and also FAD. So this is a one-cell embryo, and it's the same process for FAD. We image it and we excite it. The photons, and in this case NADH, will be excited. They will become bright, and then at some time they will lose that brightness and actually go to more of a steady state. So the data we get from each measurement is the brightness of the molecule in question, and that's NADH and FAD. We get the time that they take to actually fluoresce and to actually come down, so the photon excitement. We also can calculate the fractions bound to the enzyme. In addition to this, we can also get the redox ratio, so the intensity of NADH divided by the intensity of FAD. So we get a lot of metabolic information from these embryos. So this is an example of mouse embryos growing from the one-cell stage to the blastocyst stage. And what we actually get is actually an image of embryos growing, and you can see here on the left-hand side one-cell embryos growing, and this is their NADH profile. And if you're looking carefully, and I hope you can see this, you can see that the ones on the left are actually very bright at the beginning compared to the FAD, and then it actually switches over. So this is just the intensity, it's not the other parameters we're measuring, but if you look at the intensity, you can see one thing that we've known for many years, that if you look at the left-hand graph, and you can see the blue dots represent NADH intensity or brightness, and you can see as the embryo becomes a morula, the NADH becomes really bright. And then as the embryo goes from morula to the blastocyst stage, that brightness decreases, and you get a switch in the type of metabolism. On the other hand, the green light, the green circles, you can see they're very low at the beginning, and then post 8-cell or post morula, the intensity increases quite a lot. So you can see this switch in metabolism that we've known for quite a while that occurs in an embryo as it grows. This is another way of looking at it. This is the amount of NADH bound, or the FAD bound. Again, blue is NADH. You can see there's a lot of bound fraction in the early stages of embryo development, and then post 8-cell stage, this decreases while the FAD increases. This is really nothing new, because we've known for many years, if you look at embryo metabolism, and these are classic papers by Henry Leese and others by David Gardner, that we know the embryo in the early stages, up to the 8-cell stage, prefers to use pyruvate, so it uses the Krebs cycle, whereas in the later stages, post 8-cell stage, it switches over and it loves glucose, so it uses glycolysis. So this is also showing, what we're showing with FLIM, the change in metabolism. But we can use that to look at various things. So this is another thing that the fluorescence lifetime imaging microscope allows us to do. We can look at metabolism. So if you look at this embryo going from the 1-cell stage to the 2-cell stage, you can see the spots of white, which is the brightness of NADH here, but we can actually measure a second thing. So you can see here a purple band at the 1-cell stage. These are spindles. So one of the things we can do with the same microscopy is actually, as an embryo is developing, we can actually start looking at the spindles, and hopefully using this technology with the spindles also, we can actually see when the spindle's behaving, and possibly this would let us know if embryos are actually undergoing abnormal cleavage, if we actually see abnormalities in the spindle formation. So what we get is a lot of metabolic information, and also we can actually obtain spindle information or cleavage information that might tell us that aneuploidy may be occurring in some embryos. So how do we get metabolic imaging? What type of report do we get? So these are three 2-cell embryos on the top and looking at NADH fluorescence. The left-hand side is a classic stain used called mitotracker, so it's basically telling you where the mitochondria are. But actually, the middle and the right hand are actually NADH fluorescence on the top and FAD fluorescence on the bottom. And then we can actually look at segments, so we can tell by overlapping the images where the mitochondria are actually metabolizing, and if any metabolism is occurring in the cytoplasmic fraction. So we can actually even get to that level of resolution with NADH in particular to see what the portion of metabolism is in the mitochondria and the cytoplasm, especially for NADH. So how can we use this? So this is an example of how we've used it, and this is a study done with Embryos at Yale University, a fellow Turk for you. So this is looking at a mouse model with a problem in its mitochondria. And one of the things we did, we looked at the oocytes with problems in the mitochondria, but we also tried to look at if we could distinguish between young and old oocytes using this technology. So young and old oocytes, there's always been an argument whether mouse embryos mice go through menopause, let's say. So we're never sure. But if you look in the middle panel, it's mitochondrial DNA measurements of young and old oocytes. You can see there is some difference, but there's quite a bit of overlap between the young and the old oocytes in this picture. And actually, they don't come to significance when you're measuring mitochondrial DNA. On the right-hand panel, though, if you look at some of the parameters we use in metabolism, we can actually draw a pretty fine line between young and old mouse oocytes in terms of looking at their metabolic qualities. So this actually tells us that this type of technology may allow us to look at old and young oocytes in actually humans too. And actually that the metabolism is very key in actually how we actually may be able to pick a good and a poor oocyte. Well, we're very interested in oocytes and everything, but the reality is can we use this technology to look at human embryos? And this is some preliminary data that I'll show you now that is the characterization of phlegm metabolism in discarded human blastocysts. So we've been performing quite a lot of studies in the last 4 or 5 months, and we've looked at over 200 biopsied PGTA blastocysts, obviously discarded because they were aneuploid, the majority of them. Only a small percentage were euploid, 100 not biopsied, and 100 just discarded embryos because they were of poor quality. These are human blastocysts. On the left-hand side is NADH metabolism, and on the right is FAD. And you can see here we can get quite a lot of information about the trophectoderm, about the inner cell mass. You can see pockets of different areas that are metabolizing. So we can get a lot of information on how the embryo or the blastocyst is behaving metabolically. And this unlike the experiments I showed you before where we look at the embryo from the 1-cell stage right through to the blastocyst, this is only taking one snapshot of a blastocyst in time. So once we have that information, we can also isolate areas of metabolism. We can look at areas of the trophectoderm. We can portion out the inner cell mass from the trophectoderm, so we can get quite a lot of information. I can't show you all the data that we have because we're actually presenting that at ESHRE and hopefully ASRM in the future at the end of the year. But what I'd just like to give you is a highlight of some of the human embryo data we have so far. So the analysis of discarded human embryos has shown that non-invasive phlegm metabolic imaging can detect these 3 things. We can actually detect significant differences between aneuploid and euploid embryos, which is actually, I never thought we would have been able to do that, but it appears that we can see differences in these embryos. The next 2 are really distinct. So we can see distinct differences in the metabolic signatures between the inner cell mass and trophectoderm of these discarded human blastocysts. It is very clear that these metabolize completely differently once they become blastocysts. The other thing we see is a very large change in NADH and FAD between day 5 and day 6 in human blastocysts. So these last 2 parameters are really interesting because it tells us that embryos or human embryos are behaving very differently and we would hope that these differences may allow us to understand which embryos are viable and which are non-viable. So we're very excited about this data in the human at least. So we think that metabolic imaging and the understanding of embryos and using technologies such as FLIM will help us with viability assessment, but I think in the future it may also help us in assisting to look at how to develop media and how embryos will actually, which metabolites they prefer to use and which they don't prefer to use. So I think it'll be a very powerful technique, not just for viability, but for other things in the future. FLIM is not the only technology out there, so it's one that we've been using, but there are some amazing microscopy technologies out there that actually are giving us a lot of information. So one technology that actually is very powerful is a microscopy technology called hyperspectral microscopy. This actually, similar to FLIM, looks at autofluorescent factors, but actually has the ability to look at many more than just the two we're looking at. So it can actually make an image of multiple autofluorescent proteins or factors that are within a cell. And this is a study presented by Jeremy Thompson's group looking at bovine embryos, and they were looking at something that is very close to the heart of embryologists, the question about oxygen concentrations. And they found completely different imaging profiles between embryos that actually had grown in 7% versus 20% oxygen. So it shows you that this technology is very powerful. So what else could we look at? Again, we have this embryo that's available to us and is very amenable or very easy to look at to find something non-invasively. So we can look in the culture media, we can look at microscopy technologies, but one of the most amazing things we can look at is actually the ability to test the media around the embryo. And I'm just going to talk quickly about some technologies that are looking at the production of things by the embryo. And one of the things I will talk about are DNA, which I think you will be interested in. So obviously people have been looking at this question for many years, and this is an old paper now from Mandy Katz-Jaffe's group using a technology called proteomics to look at what factors may be in the culture media. And they asked a very difficult question about whether an embryo, an aneuploid embryo and a euploid embryo have different factors secreted into the culture media. What they were able to show that there are proteins secreted into the culture media that are different between an aneuploid and a euploid embryo. So this was very exciting, I think, at the time. They're still working on this, so this is, it takes a long time to do these types of studies, but it also indicates that there are distinct differences in how an aneuploid embryo and a euploid embryo metabolize, not just in metabolites, but actually in their protein function. We actually used a technology that was actually very powerful at doing two things. One of the two biggest problems you have when looking at embryo culture media are two, that you're working with very small volumes, and secondly, that what an embryo produces is not a bucket load of proteins. It's a very, very small amount of proteins. So if you use a traditional assay to look for these things, you have problems because of the low sensitivity and the low volumes you're actually faced to work with. So we actually worked with a company that actually has a technology to do a protein assay, an analysis using a type of ELISA assay, but actually do it at a digital level. And this technology is called Simoa. It's a technique that actually is used in looking at blood samples now. They're testing it for a lot of things like Alzheimer's, so looking for very low levels of factors in the blood. And the beauty of this technique is that they can look at femtoliter quantities in the culture media. And it basically takes the assay down to a single molecule resolution as compared to a normal assay, which looks at a much higher resolution. So this is the company, Quanterix, that we've been collaborating with. And what we wanted to do was look at two factors that may be interesting to look at in the culture media. The first was interleukin-6, and this is how this assay, this is just an interesting slide because the technology they use, if you look at this is basically a compact disc, if many of you remember compact discs, and you can see little squares around the edge of the compact disc. this is where the array takes place and that the imaging takes place. So this is an old technology produced by Sony, and they sold it to a biotech company to use it. So this is what we found when we looked at interleukin-6 expression in the culture media. So we did two series of analysis, and we looked first at embryos that had implanted, at the culture media of embryos that had implanted, and the culture media of embryos that failed to implant. And here you can see that both times we did this series of analysis with the embryos, we found significant differences in the expression of interleukin-6. You still get quite a bit of overlap though, so it still needs a lot of work. One of the other candidates we wanted to look at was HCG. So a lot of people have published data on HCG, and we thought we would develop this assay to look at HCG levels. Now, if you look at the bottom left-hand panel, one of the first things we found that this technology was so sensitive that we actually found a lot of HCG in the albumin that we use in the culture media and the serum substitutes we use in the culture media. So we actually did this experiment. We found that we couldn't actually see a difference because there were so high levels of HCG in just the base control culture media. And this technology is so sensitive that it was able to actually, it was picking up that as background. So what we did a series of experiments where we actually cultured embryos in recombinant albumin. And so this was actually purified albumin, so would not have HCG. And here you can see in this series of experiments, we found a significant difference in the culture media, in the HCG expression of culture media from the negative and positive fetal heartbeat samples that we had. So this is a very promising technology, and we're now looking at recruiting clinics that actually use recombinant albumin in their culture, because the technique, the culture experiments we did were actually with very short time when we cultured embryos. So if anyone does long-term culture using recombinant albumin, I'd be very happy to hear from them and use this technology. So this was very promising, but we actually have something that is actually even more promising, I think. This was a paper that came out now 4 years ago from Sunny Z's group at Harvard University. And what it did was show something that everyone was very surprised in. But for us, people that are very interested in non-invasive assessment of embryos, it really got us excited. So what this showed was that you could do non-invasive chromosome screening of human embryos by looking at the culture medium. And they actually did a series of experiments where they analyzed the culture medium of embryo culture medium from human blastocysts. They did whole genome amplification and next generation sequencing to see which of the embryos or the culture media showed that the embryo may be aneuploid or euploid. They transferred some of these and actually achieved successful pregnancies. When one group does this, you always wonder, okay, that's interesting, but I'm not sure if I can believe it. There's always some doubt, especially with something as controversial as this. But now we're starting to see many papers and many abstracts of factors being secreted into the culture media, especially DNA, indicating that people can actually look at actually aneuploidy measurements. So there are now many papers, there have been many abstracts published, and we've now seen a series of papers by a number of independent groups showing that this technology actually could be valid. This is one paper I've picked, and this is from Carmen Rubio and Carlos Simon from iGenomics, and they were looking at the origin of cell-free DNA in the spent medium. And they did the experiment that everyone expects. So how does it compare when you do a trophectoderm biopsy? So basically, they looked at trophectoderm biopsy results, and they looked at what was in the spent culture medium. And in this study, they only found that when the trophectoderm biopsy showed a euploid or aneuploid result, how many times did the spent culture media agree with this result? And they only found actually that only 30% of the embryo biopsies and the media actually agreed. So here you would think, oh, that's not very exciting. It doesn't look like this technique will work. But actually, many people have been studying this now, and there's been improvements in the techniques and technologies in both the culturing of the embryos and actually improving the assessment of the DNA in the culture media. And you can see here now, this is just a series of papers, and this is actually has improved even now. But you can see from very low results in the 30%, 20%, people are now reporting in the 80% range of concordance. Why wouldn't we get 100%? Well, you're looking at different things. You're looking at the embryos secreting and the whole embryo secreting, and you're comparing that to actually a small biopsy of the trophectoderm. So I would never expect to get 100% concordance. And that is actually interesting because it may actually show us that there are some weaknesses in PGTA as we currently do it by biopsy. And we obviously know that embryo mosaicism is one of those weaknesses. So this study here shows us now that we're actually improving this technology. This is another paper published by Catherine Rakowski with, in collaboration with Sunny Z, who originally published this technology. And they again, in PNAS, showed very high concordance rates when some of these technologies were improved. So I think there's a lot of hope for this technology. So currently, this was a more recent paper, and again, the concordance rates are very high, and we would hope that these types of concordances will actually continue. And you can see here, people are starting to look at a bit more embryology type things. So what about day 5 versus day 6 and 7 biopsies and embryo culture media? So here, this is showing that possibly culturing the embryos for an extra day may actually improve concordances. So you can see here from 60% to up to 80%. And I can say now that in 1 week's time, there is a study coming out by this group in collaboration with about 6 clinics around the world, looking at over 1,500 embryos, that is showing a very high concordance level of around 80% over many embryos. So I think that'll be exciting, and that will be published in the American Journal of Obstetrics and Gynecology. One of the interesting things, though, is what happens when you have a euploid trophectoderm embryo, so a biopsied embryo that was euploid. Does it always agree with the blastocyst media? We know that it doesn't. But what about the embryos it doesn't agree with? So this is an interesting, very preliminary data, and I think this needs to be confirmed by a much larger study. But you can see here there are 17 cases where the euploid trophectoderm biopsy embryo, which was transferred, actually also had a euploid agreement with the blastocyst media. And you can see here that the ongoing implantation rate was around 53%. So that's what we'd basically expect, a bit higher maybe, but that's what we'd expect. What's interesting, though, is the euploid trophectoderm, when it didn't agree with the blastocyst media, the implantation rate was only 17%. So this may be indicating that maybe looking at the secretion of the whole embryo and looking at the blastocyst media may actually be giving us better or more information that we get from the biopsy. So this is still waiting, we still need to wait to see this. So, but these data are very exciting, I think. So I think if you question me about which biomarker or which non-invasive biomarker looks the most promising currently and which one we will see in the near future, I think DNA in the spent medium is something that we may be using within the next year, I would say, in our labs. But I still hold that there are many other technologies out there that will allow us to do this non-invasive assessment. So how can we improve this technology? How can we improve this system and make non-invasive something routine in the embryo lab? And I think one other technology that lends itself to actually allowing us to do these non-invasive technologies are actually some discovery platforms. And one of these discovery platforms is microfluidics. So this was a paper published many years ago, more than 10 years ago now. And you can see this, you would never guess is a microfluidic system, but it's actually a system that allowed a lab to actually measure the single embryo and look at the culture media that came in and out of that single embryo. So basically what it said was you could do a metabolic analysis of a single embryo on a chip like a microfluidics chip. So this is really exciting, and I think this is where embryology will probably go in the future, whether it's looking at DNA in the media or whether it's looking at metabolites or whether it's actually imaging the embryo also, that I think somehow microfluidics will come into the system. And this is a review on microfluidics for embryo culture and selection. And this is quite a few years now, and this field has actually jumped quite a lot now in terms of using microfluidics, not only for embryos, but for gametes. Gary Smith has been one of the leaders in this technology. So I think in the future, this is something that we will look at. So how will we look at these embryos in the future? So this is an example of how an embryo may be cultured in the future. You will have little individual wells that your embryos will sit in. There will be fluid coming in and out of those wells that will actually be an analysis port, and actually media coming in that maybe we will be changing depending on how that embryo will be developing. That fluid will be tested in some way, and each embryo will have its own little profile of knowing what the embryo, how it's developing, what type of things it's secreting, what factors it's secreting, and also as it's growing, we will also try to understand how that embryo actually, what the needs of the metabolic needs in terms of culture media are for that individual embryo. This type of system will also allow you to visualize the embryo. So whether you're doing time-lapse, morphokinetics, or some sort of metabolite signatures such as FLIM microscopy or hyperspectral microscopy will also be available in this type of microfluidic system. So I would hope, and I think that in many years, in probably 5-10 years, that this could be the type of system that we will be using in a lab and actually allowing us to do the analysis of embryos and actually culture embryos. So how is invasive versus non-invasive? Right now, we're not that good at non-invasive systems, although I think we've really got improvement. So I think in very near future, and we've also already seen that with DNA assessment, we're getting very close to the same percentage of live births. And I think very quickly, we're going to be able to surpass that level of achievement compared to invasive embryo development. So I think what we're seeing is that in the very near future, that we will not be doing invasive biopsies anymore. And I think what we're going to be doing is actually checking the embryo either using a technology microscopy based, using culture media to test the embryo, or both. I would guess that it probably will be both. So in the future, genomics, transcriptomics, proteomics, metabolomics will all be achieved using a non-invasive technology. I think one of the biggest omics, though, is the economics. So how much this will cost. I think already we're getting an indication that actually the DNA testing will be pretty equivalent to actually what we do for biopsy, what it costs for biopsies at the moment. The difference is that you will not be spending as much time doing the biopsies, doing the actual embryology side, training people to do biopsies. So I think in some ways, actually, some of the non-invasive technologies may actually be cheaper than the invasive technologies. So it'll be interesting to see how the economics ends up panning out in terms of these technologies. So I think in the future, the technologies we will use will be non-invasive. Definitely embryo assessment will lead to a non-invasive technology, and that's because of the access we have to the culture media, to visualizing the embryo. I think that will definitely be a technology we will use. I think also the uterine testing may also be done in a less invasive form, and we may not even have to do a biopsy of some sort. We may be able to take the fluids of the uterus and do uterine synchronity using a fluid-based technique or even an imaging-based technique. And also sperm selection will actually probably be done using some sort of microscopy or non-invasive testing platform. So I think all the future transfer efficiency, so putting the best embryo with the best paternal nuclei and into the best uterus will actually all be done using non-invasive technologies. So I think this will be the future of IVF. So finally, I'd like to thank everyone for listening. I know it's difficult to do some of these lectures and to understand, so even over online to hear these, but I'd like to thank my collaborators, Daniel Niederman in the Harvard University for the FLIM microscopy, Marta Venturis and Jamin Shah did a lot of the human work. The Boston IVF embryology staff, Emily Seidler, did a lot of mouse work in this using the FLIM microscopy, as did Tim Sanchez, who's a huge help with the FLIM technology. And some of the mouse data was done with Emery Sally and also in collaboration with David Gardner. So I'd like to thank you. If you have any questions, I can do my best to answer them. So you can email me at this address and ask me the question. I will do my best to answer you. And finally, I'd just like to tell people that we are organising, so David Gardner and myself are organising a Future Innovations for Embryologists. So if you're interested in embryology and you want to hear some amazing lectures, there's free registration, so you're very welcome to log in and participate in this conference. So thank you again for listening.
Denny, can you hear me well? Oh,
yes,
so there was an audio problem in the beginning, in the introduction part, I think. So my colleagues reminded me, I mean, after some time, so I couldn't, I think, give you a good introduction for you, but everybody knows you. So you are very well known, a colleague of ours in the international area. So I think we have a couple of questions, and they're still coming up. So, so I'd like to take an advantage of asking the first question from my side. I have actually a couple of questions to you. The first one is that, so you started your presentation with discussing about AI, so how we can use artificial intelligence to select non-invasively the embryos. I also know that you are working intensely on sperm side. So how far did we achieve for sperm selection by artificial intelligence so far?
So probably less with AI is probably being used less with sperm selection. I think it will come though. You know, obviously, and I think Turkey is probably leading the way in some of the microfluidic studies. So I think a lot of people are interested in microfluidics with sperm. AI is actually, where it is actually helping is looking at not just morphology, but motility patterns and trying to recognize which motility patterns actually are the ones that were very interesting and maybe related to the successful sperm. So I think where artificial intelligence is helping with sperm is probably in looking at more understanding better the motility patterns. And I think eventually all this data will come together, looking at motility, morphology, and maybe even, you know, the simple sperm concentration assessment we're getting. So I think AI, even though it's sort of probably behind the, you know, it's not as advanced as what we're getting with the embryo, I think at some stage artificial intelligence will also be used for sperm selection.
Okay, so we have a question from Leila. You know Leila very well, from Yale. Yale Times. Hi, Denny. How would we make sure that we also measure cellular autofluorescence from some other molecules, not just only from NADH and FAD? What are the limitations for this technology?
Well, so the limitations, so at the bandwidth, the fluorescent bandwidths you're looking at, so the fluorescence lifetime imaging microscopy, the way it's set up in our hands, we're only using the wavelengths of interest that where NADH and FAD are actually fluorescent or active. But something like hyperspectral microscopy is much more powerful and allows you to look at a whole range of autofluorescent factors, whether it's a protein, a metabolite, or any factor. You know, even things like some of those cytoskeletal filaments are actually autofluorescent. So there are a range of autofluorescent factors that you can look at in an embryo or any cell that allows you to look at this. So the limit is governed by the bandwidth or the wavelengths that your microscope can look at. And some of the hyperspectral microscopy microscopes are actually able to look at 9, 10, 11 different autofluorescent factors. So if you're interested in this, I'd urge you to look at the paper by Jeremy Thompson and some of the other studies they've done, which are really fascinating.
So we should be studying quantum physics, to get some expertise in non-invasive selection.
So, I always tell people that if you're interested in these types of studies, don't sit in your lab and try to do them yourself. I think a lot of these sort of technologies are available. And, I'm sure in all the Turkish universities, there are many people working on many interesting things. It's a matter of going out and talking to people and looking at, what are people doing in other fields? So I think a lot of the things we learn of, even the genetics that come from the cancer field. So I think just looking at physics, looking at, molecular biology, looking at biomedical engineering, all these techniques will help us go forward in what we do too. So I think you're right. Talking with physicists, you may not understand each other, but you can get a lot of information. The group we work with actually to do the FLIM is actually in the physics department. So I think this is actually one thing that I would encourage people to do if they're interested in this type of research.
Thank you. The next question is the ploidy status of the trophectoderm and inner cell mass are concordant? What if the aneuploidy of the TE is not so uncommon in babies born healthy? May a cell-free pre-implantation test help us to test better in this manner?
So I would think that by looking at the whole embryo, so doing something like DNA screening of the whole embryo may actually give you more information. The trophectoderm biopsy, although it's a very powerful technique, but we all know that when we take 5 to 6 cells from a trophectoderm, it may not always represent the rest of the embryo. And actually, in the paper that I mentioned that's going to come out in next week in the American Journal of Obstetrics and Gynecology, there were a series of experiments looking at inner cell mass and trophectoderm and looking at the concordance of those. But there have been many studies that people have looked at, and quite a few papers that have been published in Human Reproduction, Fertility, Sterility, looking at this question. In general, the concordance is quite high between the inner cell mass and trophectoderm, but it's not 100%. So we know that when you're biopsying the trophectoderm, that you are taking a risk at some stage that sometimes that trophectoderm is not representative of the whole embryo.
Yeah. So in a similar question, in a similar type of question, actually, from Chilar. She asked, regarding DNA fragments in the spent culture media, are those specific DNA fragments that could be transcribed or just random DNA fragments, so neither introns or exons?
So asking about the sperm DNA.
I think cell-free DNA in the spent culture media. I think she's asking the... Yes.
In the blastocyst culture media. So we're actually doing a series. I hope this is going to answer her question, but she has my email, so she can ask me if I don't answer it. We're actually doing a series of experiments now to try to understand the origin of that DNA that is secreted by the embryo. So we're trying to understand where that is. So what is interesting, though, is that if you take DNA from embryos that were post-morula, so you take them from, you actually take media from day 4 to day 6, it's much more efficient in giving you true information about the euploidy or the ploidy status of the embryo. It looks like if you take from day 1 to day 6, for example, that DNA in the culture media isn't as efficient at telling you the true value of that embryo. Now, whether that DNA, where that DNA comes from, we really have no idea yet. You would guess that it's some sort of signaling mechanism to the uterus. People feared that it was like apoptotic DNA or just fragmented DNA, but it doesn't appear to be the case. It appears to actually have some value, let's say. Now, whether that value is in actually some crosstalk between the embryo and the uterus or within the embryo itself, we really don't know yet. But I think in the coming years, I think this will be a really interesting area of research and knowledge that we should be getting.
Okay.
So I hope that answered her question.
She has even a longer question that I'm going to translate to you later. Again, I'm continuing with the NIPT, non-invasive PGT-A actually. Professor Yaralı asked a question regarding the same area. So we know that in order to get the proper sample for non-invasive PGT-A, you have to culture the embryo until day 6 and day 7. And assuming that you have a beautiful embryo on day 5, like Gardner score 5AA, would it be okay to culture this embryo to day 6, 7, just to get this non-invasive result? Is it justified?
So we just did we were part of this clinical trial with iGenomics that's going to be published. And we had that fear ourselves. So we shifted, for all the cases where we were doing non-invasive PGT, we shifted to actually biopsying embryos to day 6, but also collecting the media at day 6. Even though there was some resilience by the embryologists because they were scared of leaving embryos from day 5 to day 6. In the end, we were fine. We didn't have any sort of higher rates of damaging the embryos. I don't even know if we had any damaged embryos after biopsy. Our no result rates were very, very low, much lower than what we normally have, which was interesting. So overall, we didn't experience any of these problems that we thought we would have. So and that includes actually allowing embryos to hatch. Now, obviously, sometimes it's harder to biopsy those embryos, but actually, after a little bit of time, all our embryologists were quite comfortable at biopsying on day 6 and not having any issues. So I think this is a matter of training, and I think, hopefully confidence that you can get a good result out of the non-invasive technology.
So you didn't have a significant degeneration rate after you cultured them to day 6 and day 7?
No, not at all. We actually, so this is with absolutely no proof at all, but what was interesting, our euploidy rate seemed to be higher when we switched to biopsying or to looking at the embryos on day 6. I don't know if it was just coincidence, but we actually had I think we had some positive benefits to it rather than negative. I can definitely tell you that it appears from the data that if you do a day 5 biopsy or you take the media before 30 hours of culture, by leaving it from day 4 to day 5 and only have about 30 hours of culture, your ability to get information from the DNA drops significantly. So it's a balance, if you want. I think if you're doing this technique initially and you're setting it up, you may actually want to do some embryos on day 5 and maybe leave some just to be more comfortable. And like I said, I think our embryologists became quite quickly quite efficient at biopsying all the day 6 embryos, regardless of their stage, if they'd expanded a bit more than what we were used to.
It's a very important point because one side, you have a new technology that you would like to adapt your system and take the advantage. And the other side, it really challenge your current, let's say, application because, you are not used to culture the embryos to day 6, day 5 in a routine way. And I'm going to try to get your opinion in a similar fashion for microfluidics and spent culture media assessment. Because, for example, for NIPT-A analysis, you have to have a certain amount of DNA in the spent culture media. So you have to culture the cells in a static and a smaller volume environment. But if you want to adapt your system to microfluidics, it creates a little bit of controversy. Right? I don't know which one's going to be the winner in the end.
You're right. So you will have a balance where you maybe you cannot use all the capabilities of microfluidics to do the testing. So you will have to keep maybe the media static. All I can say is that I would say by the time we are doing this maybe more active culture, you know, just think how quickly the molecular biology side has moved in terms of actually being able to assess very, very small amounts of DNA. So I think that, you know, we may actually, that 30 hours, 40 hours of culture that you need, I think very quickly may come down to even, you know, a handful of hours, you know, 5 hours. So I think the molecular biology techniques in terms of amplifying the DNA and getting efficient information from it, I think is changing also very quickly. So I would think that maybe in a few years and even less, that question may not be as pressing. And, you know, maybe we will be able to easily assess embryos on day 5, firstly, but then if we're using a microfluidic system, maybe even a few hours of culture will give us enough DNA to get information.
Again, in continuation of this area, Murat asks whether it is possible to characterize nuclear and mitochondrial DNA simultaneously in spent culture media. So
nuclear in spent culture media, no, I don't. Not that I know of. No, all this. So I don't know if we can. Basically, when you're looking at the DNA, you're amplifying the whole whatever's in your culture media. I'm not sure if some of the data they've been able to obtain allows you to also look at mitochondrial DNA. That I think would be a question for the people doing the actual diagnosis. I don't know the answer to that, but my guess is probably not.
Okay. Chilar's second question is about the film study, so I'm just saving it, just to finish with the non-invasive spent culture media questions. You know that there are several protocols for embryo biopsy. Sometimes you have a laser shot on day 3 and wait the embryo to expand on day 5 and day 6, or you can just wait until day 5 and go simultaneously for laser and the biopsy. Do you think that the way of biopsy important for non-invasive studies? I know that iGenomics protocol requires no touch of the embryo at all before day 6 and 7, but what do you think, and what is your personal opinion on this?
So we were doing the iGenomics protocol. So again, you know, this is really challenging the embryologist. So we routinely used to biopsy, laser hatch the embryos on day 3. And then obviously we would, on day 5, day 6, or sometimes even day 7, we would biopsy them. So when we were asked to do the iGenomics protocol of leaving the embryos and not lasering them, we had a revolution in our lab. Basically, people were very scared. But it's like I said before. So basically, what we were doing, we were collecting the media and then doing the biopsy. So all the samples we collected were with non-assisted hatching or non-lasered embryos. So, and like I said before, our embryologists became pretty efficient at biopsying at day 5 or day 6 actually, and doing the hatching then. So it really you know, it's a matter of being able to be flexible and sort of, you know, have well-trained embryologists do this technique at least at the beginning. So for us, it really didn't create a problem at the end.
Okay. So I'm coming to Chilar's question regarding film studies. She asks whether do discarded embryos, I would say bad embryos, lag or entering glycolysis or switch their metabolism, OXPHOX or glycolysis. Moreover, if we can discard bad embryos by their morphology by our eyes, how would film discriminate with a very good morphology related to the metabolism? Is it promising? It's a long question.
So the human embryos we've looked at, and we've looked at all the majority of discarded embryos we looked at were actually embryos that were actually very good quality blastocysts that were frozen and discarded because the patient had become pregnant, or they were actually embryos that were aneuploid. So in our data set, we had 150 PGT embryos, and like I said, to be biopsied, they have to be a very good quality. 17 of those actually, I think, or 20 were actually euploid. We actually had euploid embryos discarded because the patients were not using them anymore. So the majority of the data we have are actually from very good quality blastocysts. And then we have about 100 that were actually discarded because they were, not sufficient quality to freeze. So we're pretty confident that the data we have is not representative of poor embryos or poor blastocysts. We're quite confident that the data we're seeing in terms of being able to see differences between the inner cell mass and trophectoderm and between day 5 and day 6 embryos, blastocysts, we're very confident that that's actually the true image or metabolic state of the embryos, and we're not looking at just bad embryos.
Okay. We have two very similar questions, and it's a very speculative questions. It's about can genetic disorders be repaired, especially in patients older than 40 years?
Be repaired. So I don't know if they're talking about some CRISPR technologies or
chromosome transfer.
This
or chromosome transfer, maybe.
This is
it's very speculative and
really in the future.
So
this, this is interesting. I think CRISPR technologies. There are genetic techniques that maybe would allow this to happen. There may be stem cell techniques that might allow some of these to be corrected. But I think these are quite a way away, I think, in terms of being adopted and actually just shown to work, even in other mammalian embryos, I think.
So we have 2 questions regarding PGTA. As far as we know, most meiotic errors are translated by the oocyte to the embryo. Is polar body 1 and 2 testing for ploidids still remain as an alternative? Do you think?
Apart from religious grounds, because of the certain laws or ethics in a particular country, I really don't know many groups practicing, polar body biopsy. It wasn't the easiest thing to do. So the first polar body was probably easier to biopsy, but then once you get to the second one, it was always more difficult. A lot of times they would fragment. So it was actually, I haven't done that many, but the ones I did, I never found that easy to do. But I really don't know of many countries or many laboratories that are doing it still.
Professor Yaralı asked whether the STAR trial had an impact on the policy of PGTA at Boston IVF in real life.
So we're an interesting clinic because we still only do about 35% PGT. So we're not one of these clinics that do 100%. So I'm not sure. So Boston or Massachusetts has health insurance for IVF that the government gives. So the majority of our patients, probably 80% of our patients, actually have an insurance. So this sort of changes their mind about doing PGT, I think, because they have coverage anyway. So for us, it didn't really change anything. And we've published, even before the STAR trial, looking at our own data, we've published papers showing really that if you're under 38 or 35, the benefits of PGT aren't that dramatic. So it really never changed our practice anyway. The majority of our doctors were really using it more for the over 38 patients, and less so for the 35-year-olds, let's say, or younger.
I have a question. In one of your slides, you mentioned several studies that you've done with Quanterix, the company, and then you had came up with the two informative markers. One is interleukin-6, and the other was an HCG. I mean, HCG can be understandable, but what do you think about interleukin-6?
So, one of there are two reasons we chose interleukin-6. One is because the company already had the assay set up, so that's sort of it sounds stupid, but it just sort of, we were testing the principle, but there are quite a few publications looking at uterine secretions, and actually some fewer publications on embryo culture media indicating that the interleukins were candidates. So we actually looked at IL-1, IL-6, and IL-11, I think, or 10. So we looked at a few candidates, but to be honest with you, one of the main reasons we chose interleukin-6 was because the assay was already established by the company.
Okay, that's a quick answer.
Sometimes the reason we do these things are not just through main intelligence. There are other reasons.
So what do you think about blastocelic fluid analysis for genetic testing?
We've never tried it, to be honest, so I really have no experience with it. The people that I've spoken with that have done it, obviously some of the Italian groups have published some very good results, but I've spoken to other groups that have found it difficult to make it work. I don't know the technique that well, but I've heard conflicting results about its utility.
Okay. Again, regarding non-invasive single blastose culture media, what do you think about the mosaic embryo status? I mean, can we determine the mosaic embryos with this technology? And what about single gene disorder detection?
So I think the Chinese group actually have done now some cases with single gene mutations. So I think for the single gene mutations, I think it is possible. I don't know. I think people will be careful about using it, and I would believe that biopsy will still be the technique used just for now for doing mutation analysis. In terms of using, can we look at culture media and look at mutations? I think we still have to wait for that to be probably more comfortable with the analysis with aneuploidy before we start going to to things like mutation analysis. But it appears that we do get enough DNA to do these types of analysis. In relation to mosaicism, mosaicism's a sort of byproduct of the biopsy, I would guess, if you want to look at it like that, just because we get 5 cells and maybe one of those is giving you a different result from the other 4. Now, in the culture media, you're really getting a bigger picture of what the embryo is secreting in terms of DNA. Whether we're going to pick mosaic embryos, I'm not sure how they're going to present the data, because in effect this is going to be an algorithm of some sort, bioinformatics, that will tell you how that an embryo is euploid, but there will, I'm sure, be DNA from the culture media that will sort of veer from the euploidy status, and people may think that it might be an aneuploid or mosaic embryo. What I think will happen is that say you've got 5 embryos that you're testing, you will probably be told that embryo number 2 and number 3 are the ones that you should transfer, but maybe number 4 is, we're not as sure about, but, you shouldn't throw it away, but you may want to keep that for later. And that may be sort of the embryo that is thought to be mosaic, or maybe has a slightly lower chance of being euploid, let's say. So I think we may see the data presented in a different way, I would say, than what we're currently seeing.
Is there a specific technique to minimize cumulus cell base contamination for this non-invasive testing?
So actually, it's an interesting question, because we, in the multi-center trial that iGenomics did, we were the only clinic that still does IVF or inseminations. So we do about, probably about 50% ICSI, 50% routine inseminations. So we actually, in the groups that, in the samples we gave, we also had non-ICSI samples or insemination samples. The results were exactly the same as ICSI. The only thing we were very careful about was washing. So when we moved the eggs from the cumulus cell samples, we were very careful washing them to the next culture media. And actually, for the day 4 change that we were doing, we actually introduced an extra wash to actually make sure that we were not contaminating them. And in the end, our contamination rates, all the no result contamination, whatever you want to call them, the rates were exactly the same as ICSI compared to our IVF samples. So the non-invasive technology can be used for IVF samples also.
Okay, and the next question. So when span blastocyst media is aneuploid and trophoblast biopsy is euploid concomitantly, there is still about 35% chance of pregnancy. So in future, can we totally discard aneuploid embryos reported as such by span blastocyst media?
So, this is this is the question that everyone asks, even about biopsy. So, I think that's why I think what will happen, I think, in the future is that these techniques will probably rank the embryos as, you should transfer it, you may want to think about transferring, or don't transfer it. And that may depend on the analysis you get in terms of the chromosome analysis, whether, your clinic may decide that it doesn't want to take that risk. Other clinics, just like now, will take the risk of transferring mosaic embryos. So I think that'll be something that different clinics will deal with differently. For example, our clinic, we don't ask, we only ask for a euploid analysis and aneuploid. We don't ask our providers to tell us if there's any mosaic embryos. So we don't have to have that discussion with patients. So I think it'll be something that each clinic will deal with differently. And also, some countries you can't discard these embryos. I think Italy still, you're not allowed to discard aneuploid embryos. So I think different countries will deal differently with this type of information.
Right. Next question is, although maternal side is perfect, what do you think about the embryos which has no aneuploidy and no pregnancy?
So these are the real difficult cases. So if you were talking with Carlos Simon, he would tell you that it's the uterus. If you're talking with me or someone similar to me, I would tell you that it could be the sperm. So, and I think the answer is both. That sometimes it could be the sperm, and I'm positive many times it's also the uterus. So, these are very difficult cases when you've got, multiple euploid embryos, you transfer them back, and even, if you think the uterus is fine, you don't get a pregnancy or you get a miscarriage. So I think that's the type of patient that you start thinking about, doing an endometrial test, or even, using something like microfluidics to prepare the sperm, or PIXI. I think, these are the difficult cases, but that's when you start, looking at a possible sperm factor or a possible uterine factor, I think. And there may be other factors too that we just aren't able to diagnose at the moment.
If we look at the literature, current literature, we have that there are some metabolomic studies focusing on the general health of an embryo. I mean, the biochemical pathways. But do you know any biomarker, or do you expect that we're going to have a biomarker which also who's going to tell you eventually about the aneuploidy status of an embryo? I mean, simultaneously,
So I presented some data saying that that using this slim microscopy, we're able to see differences between aneuploid and euploid. So the problem there, though, is that I can't tell you it's a chromosome 21 anomaly or a chromosome 16. Our initial data indicates that it's harder it's very, very difficult to look at the individual chromosomes, but we can look at the population. Now, I think some of the proteomics data may help in telling you the types of aneuploidies. So I think there may be evidence to indicate that you could look at using a factor, a protein, or a metabolite that will tell you aneuploidy or euploidy. I would find it very hard to think that you could actually give a chromosome analysis, if you want, by looking at such a factor. I hope that's answered the question.
I think, yes. Yeah, and one of my colleagues says it's out of topic, but will you please share the tips of using routine insemination? You said that you had a lot of IVF in Boston.
So we routinely, and it's mostly because of insurance. So if a patient has less than 4 million motile sperm in their preparation, the insurance will pay for ICSI. If they have more than that, we basically do IVF on them, because they will have to pay for ICSI by themselves. So a lot of patients don't, when they're getting something for free, they do not pay. They will not pay. So we still do, like I said, half of our cases, probably more than half, a routine insemination. So we're not a clinic that does 100% ICSI. So for us, it's just been normal that we do this. So when we do the analysis, we do see some differences in IVF and ICSI, and some of those have been published. So we do see, I think cumulatively possibly better, a few more embryos per patient, and there are some small differences in pregnancy rates, but it's very hard to do these studies. But we definitely don't see any benefit of doing ICSI as 100%. We do, we probably do see more fertilization failures than, a clinic that would be doing all ICSI, but the majority of the fertilization failures, if we look at patients with a lot of eggs, we don't see a difference, but you do, if a patient only has 1 or 2 eggs, for example, that's where we normally see them. So because we've always used this technology, it's hard to share tips. It's something we've been doing routinely. So, but that, like I said, our cutoff is around 4 million sperm motile sperm per patient.
Okay. So I'm just having the last 2 questions. The first one is, how close are we to carrier screening on the embryos?
Well, there is you can do that. Even now, it's just a matter of the resolution of DNA you get on each embryo. There is one company in New York that is giving you not exactly carrier screening, but gives you a risk analysis of of genetic abnormalities or the risk of having an abnormality. The data is not that strong, and there are some papers that look at this mathematically and will tell you that it's a very it's basically a guess when you're doing this type of analysis. So I'm not an expert in that field, but I think we're quite a way, even though there are people offering this or talking about it. I think for maybe some diseases it may be easier, but for a lot of things it would be very, very difficult to do carrier screening. You know, the classic is, you know, if your child is going to have no hair or bald or something, even in males, if you do full genetic screening, it's very hard to predict this. So I think with an embryo it might be even harder. But I think people, I think it's still quite a long way away.
And the last question I'd like to ask, because you nicely overview the AI on the current status on IVF. But I mean, what is your personal opinion? Because I started to think that although it's very challenging, I mean, it's the future, but I think we are really pushing too hard at a too early stage. I mean, because I mean, you can have a model for everything, but it doesn't necessarily to fit everything, you know? So what is your first?
Exactly. If you talk with the people that do artificial intelligence, and you know, we're working with quite a few groups doing this, they can mathematically model anything you want if you want. It's so, you know, the test is actually doing, you know, a predictive trial, and I think we're all very excited about the data that we see with artificial intelligence. I really think that, you know, the data is quite good, and I think especially you know, time lapse and morphokinetics, I think is something perfect for artificial intelligence, and so I think it will be part of that system in the future. How efficient it will be a very different question, I think. You know, the 93% really very, very good, but you know, I think if it gets to, you know, even 70%, people will start looking up and be sort of interested in this type of technology. But I think there's a lot to be said for the artificial intelligence and you know, the ability of people to do this type of thing. So I think it will be a technique and technology that will come into many things that we do in the lab in the future. Just as we see now in our lives, a lot of things we do, artificial intelligence is part of. You know, if you're using your phone, it's there. So I think it will be part of the lab in the near future in different fashion in many aspects of things we do.
Excellent. So, Denny, on behalf of Turkish Society of Reproductive Medicine and Turkish Clinical Embryology Society, we would like to sincerely thank you. Thank you for your valuable time. I know how busy you are, and I mean, you really helped us a lot in this presentation. And thank you for everything. And hope to see you soon.
Okay.
Physically in Turkey soon, hopefully. After this time is over.
I really hope so. So you promised me some kokoreçi and patsa. So when I come next time, I will have some.
Yes.
Okay.
Thank you so much, Denny.
Thank you. Good night, everybody.
Herkese çok teşekkürler arkadaşlar katılımınız ve sorularınız için. Bir sonraki webinarda görüşmek üzere. Hoşça kalın.